My name's Israt Alam. I'm a senior scientist within MIPS, and it's my absolute pleasure to chair the next session on immunology, featuring two amazing women from our molecular imaging field. I'll start off by introducing Dr. Michelle James. She's an assistant professor in the departments of neurology and radiology within the MIPS program. And today she's going to be talking to us about illuminating immune responses in CNS diseases using PET.
Okay, thank you, Dr. Israt Alam. I appreciate that. Lovely introduction. Hello, everyone. So, just while they're bringing up my slides, just wanted to start by saying yesterday was really beautiful. For those of you that were here, thank you so much for joining and just reflecting on Sam and Aruna. I spoke a little bit yesterday about all of the ways that both of them have impacted my life personally. A little bit about Sam and how he's impacted my way of thinking about science. Three things that have really stuck with me before I dive into some of my research. One, he used to say, just always start with the patient in mind. What are the biggest unmet needs? Which sounds obvious, but he'd encourage us to go speak to clinicians and say, what are the things? What do we need? And to not shy away from tackling those big problems and to think about the patient as you're developing your solutions, not to make some really long, complicated scan that's going to be really tricky for folk to participate in, but to really be thinking about how to make things practically accessible and just a possibility for patients to participate in. Secondly, he would always say, we're in the business of research, not search. And that really stuck with me because we're not just about looking once and that's it. It's a long and involved process of going back to the drawing board, about being excited to continue looking, continue getting better, and to not just do iterative science, but to think boldly. And he would always say, we're not here to do Me Too science. We're here to think outside of the box, to do big things that are going to change the field. So that's another thing. And then the last thing is really just thinking about getting things to the clinic quickly. Not spending forever in mice, but finding streamlined ways to test things out in the clinical setting. So I'll touch upon those things and how Sam really impacted my way of thinking since starting here at Stanford in 2008. Okay, so my lab is really interested in trying to understand more about the role of the immune system in initiating and progressing in the progression of brain diseases. And so why is that? Well, really over the last couple of decades, immune dysfunction and neuroinflammation in particular have emerged as these key pathological drivers of many diseases that affect the brain. And I'll show a couple of them here. We've already heard from Dr. Molmino about neurodegenerative diseases. There's a strong neuroinflammatory component that we know is involved in driving diseases like that, in addition to Parkinson's and other neurodegenerative diseases. Psychiatric diseases is another area where I think there's a huge unmet need to understand what's driving a lot of these conditions. One of those areas is certainly immune processes. Autoimmune diseases, and I'll talk about MS. You've already heard from Dr. Cleland about MS and other autoimmune diseases. Head injury is another area, and I'll never forget seeing a brain scan from the group at Hopkins looking at football players after just one concussion. You see the brain lighting up after receiving an inflammation PET tracer, and that really scared me. And yeah, makes me not want my kids to go into football. But yeah, what's going on there? We don't know. So I think having these tools to understand and follow folk with these various conditions will allow us to hopefully readjust behaviors or have early interventions. Infections is another one. Long COVID is something that you're probably quite familiar with. But all sorts of infections can trigger inflammatory responses that can persist and cause a lot of issues. Brain tumors, we just heard from Dr. Lim about that, and all of the immune components that are driving various aspects of the pathology. So how do we take a closer look? Well, that's what I've been very interested in. I've been very interested in looking at all the different cells that are involved in this complicated orchestra of just the immune cascade that it's just quite intricate. And where do you even start? Well, we've just begun, and I actually presented some of this to an immunology group not long ago, and I said, please don't judge me. We're thinking about it very simply at this stage, I think we're just starting to scratch the surface for the kinds of markers we can start to look at. And hopefully in time, with wonderful collaborators like Dr. Levine here, we can start imaging multiple targets at the same time and start looking at more complicated ways of understanding the immune phenotypes of these various cells. But here's just a little table pointing out some of the traces and their targets that we've been working on or that we're sort of looking at and thinking about in the field that we think are really helpful, at least for starting to understand the bigger picture of what's going on. And I'll talk about some of these today. I'll start with TSPO, which is looking at a marker for activated microglial astrocytes, and it's on a number of cells, but I'll get into that in a little bit. And then some of the newer markers we're looking at. I did want to mention, although I won't have time to talk about today, there's a lot of great emerging imaging agents for T cells. Dr. Wu will be speaking about that next, and she's really the authority in the field on imaging those sorts of cells. And Dr. Alam, who's chairing this session, is working on a lot of T cell activation agents. So I think that is a fantastic and super powerful area of research too. I'll also chat a little bit about our B cell work. So just jumping in, I started off in this area in 2004, developing imaging agents for TSPO. So it's been around for a long time, and we thought it was exciting as sort of an entry point, just as the initial way for us to start looking at some of these inflammatory cells in the brain. And so we made some traces. The nice thing about this target is it's expressed at very low levels in the healthy CNS, and then it seems to jump up in its expression. As soon as there's something going awry in the brain, this marker seems to go up on predominantly the glial cells, but it's also on endothelial cells, so there are caveats to interpreting these images. So I'll just state that immediately. So I've been working on this for a while, and what's interesting is I've been sort of reinvigorated with the use or the sort of the potential utility of traces like this to help us to start to get at what's going on in a number of diseases. One of these diseases is ME/CFS, or chronic fatigue syndrome. Now, I was approached by a very passionate young researcher in the infectious diseases division here, knocking on my door. She tried to email, she tried to text, and she came and she said, "We need to try this. I really think that there's such a huge need for these patients for us to just see if there's anything going on at the molecular level." And it seems that inflammation is a key factor. And so what's happening in this disease is that folk who were once really active, there's lots of stories of young folk that will just all of a sudden be taken down by this and be bedbound. They have unrefreshing sleep, extreme fatigue, muscle pains and aches, brain fog, a number of symptoms where it's so debilitating to the fact that they just can't do their day-to-day activities anymore, and it's very sad. I've been able to interact with a lot of these patients now, and it's a very tricky situation because a lot of the tests that we have available just show that they're fine. Like the blood tests are negative, MRIs are negative, and so for a long time doctors have said it's in your head, and obviously that leads to depression and all sorts of other issues, and there really just hasn't been a lot of hope. So we thought, let's see if the potential cause is a virus that could trigger sort of this long-term unresolved inflammation that then really fuels all of these symptoms that I was just talking about. So we thought, let's take what we have, which is a TSPO tracer, DPA713 in this case, which I worked to develop in Sydney with Michael Cassio and others. So we brought that here to Stanford, and actually some very wonderful radiochemists here, Dr. Bin Shen in particular, helped to set this up here. Dr. June Park has been an absolutely phenomenal force in C11 chemistry, and Dr. Bo Zhang is continuing this work here, and we really appreciate that. So we set up the chemistry here. We're looking at moderate to severe patients, so we look at the MFI, which is multifactorial fatigue index, and then we're looking at age and sex matched controls, and we're using the PET MRI. So here's some of the initial scans, and you can see already in that left image there's some hotspots in and around the cortex, and in particular in the pons and thalamus. So that was already interesting. We're like, okay, there's definitely something happening there, and we see this time and time again. But what's interesting is when we look below the head, we started to see this signal in the salivary glands, and I thought, what is that? And we started to look into the literature and talk to the infectious diseases division. They said, you know what? When we take saliva samples, there's viral load for some people, and there's also mast cells, an increased number of mast cells. And TSPO is expressed on those cells. We thought, okay, that's interesting. What else are we missing? So we're injecting these people with radioactivity. Why don't we just put them in the scanner and scan the whole body? And we don't have a whole body imager yet at Stanford. It's coming. But I said, let's just try it. And then folk were like, well, it's carbon-11. I don't know if that's really going to work with all the bed positions, but I said, let's give it a go. So that's me. I said, I'll jump in the scanner first and give it a go, because I don't want to put patients through all of this if it's not going to yield an informative image. So I went in there, and these are my whole body PET-MR images using DPA 713. And you see a lot of activity in the heart and in the gut. That's normal for TSPO expression. It's on the myocardial cells and some other cells in the periphery in the liver and gut. But otherwise, it's pretty quiet. And we thought the signal's good enough to at least try to see what we're going to see. And so these are the first images, and actually we've got 6 pairs now of ME/CFS patients. And if you have a look on the left, you can see something really interesting, that in the bone marrow, in the vertebra, it's really lighting up compared to the control, and as well as in the muscle. So if you look at the arm muscles and the leg muscles compared to the control, there's certainly more signal, and we've been quantifying that and trying to understand what's going on. Now, 2 things here. One, I showed this at a couple of ME/CFS conferences lately, and the patients are just floored. They've been writing to me, coming up, they're like, thank you just for showing us this is not in our head, there's actually something happening. We feel like there might be hope for therapy, or just knowing that there really is something going on is giving us just a lot of, just affirming us, right? So I think that's number 1. Number 2, there's been some papers that have come out recently about long COVID and immune responses that are occurring in muscle. So they're taking muscle biopsies, and there's a lot happening in the muscle that we've been ignoring or maybe missing. And so this has opened up a whole other area, and we're continuing to collect data here, and we hope that this is just the beginning of trying to understand more about these potentially viral-triggered long-term disabling diseases like ME/CFS and long COVID. So this is just our initial look. We've got more participants scheduled. We're really trying to increase the sample size, of course. It's not easy to get these patients in and everything scheduled and everything together, but we're doing it, and we're trying to shorten the scans and trying to look into ways to improve things. And we're excited to do sort of total body imaging once we get different scanners as well. Looking forward to that. We're also in parallel looking at identifying more specific biomarkers, that idea of research. Like, we're not stopping at TSPO. TSPO is fine as sort of your gateway drug into inflammation imaging, but we believe we can do better, because obviously that particular biomarker is also expressed on endothelial cells and other cells. So it's a good start, but what else do we have? So we've started now to look at a marker called GPR84 that seems to be much more specifically expressed on just your microglia and macrophages. So you can see here from the Barris Lab RNA-seq data set, this is just one example. TSPO is expressed on a lot of different cells that are found in the CNS, including your endothelial cells and astrocytes, whereas GPR84 appears to be really selectively expressed just on your microglia and macrophages. And in our own hands, we always want to validate this. We don't want to just trust what we see in the literature. We had a look at human monocyte-derived macrophages. We stimulated them with LPS. This is work done by Sydney Nagy in my lab. And we saw that, interestingly, that GPR84 really goes up, which is great. But we were seeing that TSPO actually doesn't go up in expression, like the fold change doesn't go up. And that's been reported in the literature a number of times now, that actually when you see that increased signal on a TSPO PET scan in the clinic, it just represents more microglia and macrophages at the site, not that TSPO is going up per cell. So here we're hoping that with GPR84 we could get a higher signal to background because we're actually getting that elevation in signal. And this is just to show you it's not just in the human scenario, but also in mice, so that our translational mouse to man work would be helpful, is that after administering LPS IP to a mouse, we see that really nice fold increase in GPRD4 as well. So we've been developing a number of PET tracers for this target. Dr. Mossam Khalida in my lab has been working very hard on this, as well as Dr. Isaac Jackson. And we now have 3 carbon-11 molecules and a few F18 molecules that we'll talk about. This is just one example. We've been looking at evaluating these radiotracers in cells. In this case, we're just showing hex cells that are artificially have a stable transduction of human GPRD4, and we see great specificity. We've been looking in mouse models again, just starting with LPS, starting very simple, but I think very encouragingly what we're seeing is low uptake in a healthy brain and increasing uptake as the level of disease severity, in this case sepsis severity, goes up. So it could be very helpful in staging the amount of inflammation that's present. And then we're always doing autoradiography because we love that. We get to see with more detail in the brain what's going on. And here we're showing specificity. So we always want to show specificity for our target. And just to show you a little bit about the recent work in F18 tracer development, because we always want to be moving towards F18. This is one of our newer tracers that Dr. Khalida has developed in my lab. And we're looking again at the human monocyte-derived macrophages because we want to make sure we're seeing things that are relevant to the clinical setting. And we're seeing a nice increase in tracer binding to cells that have been stimulated with LPS versus blocked cells or untreated cells. And this is actually, I don't know if Dr. Lim's still here, but just a shout out to the collaboration with him and his lab. We've been looking at a brain metastasis model. And here, because we kept on seeing in the literature, GPRD4 is very much associated with tumor progression, and it's a prognostic marker. So here we're actually showing, without any treatment, a really nice signal, and post-treatment, that signal goes away. We're also showing an EAE. In our mouse model of multiple sclerosis, we can see really nice signal in the spinal cord, so we're able to track those cells. So we're now moving towards the clinic, and I hope, just in the vein of what Sam would be trying to do, trying to get things into the clinic quickly. So this is our timeline. It's quite ambitious, but we're getting there, and we're about to do our tox studies and hopefully move towards the validation runs with our best tracer, so we're comparing them all. And then I'll just touch quickly on TREM1 and B-cell imaging, just very quickly. So we've been looking at TREM1, triggering receptor expressed on myeloid cells type 1 in EAE and other diseases. This marker is very specifically on your peripheral myeloid cells. So it's not really on the microglia at all. It's allowing us to visualize your peripheral myeloid cells, particularly macrophages, and in some cases neutrophils, trafficking into the CNS. So you can see the top left image, not really anything going on in the naive, but in pre-EAE, you start seeing that signal in the spinal cord and bone marrow. And then as the disease goes on and those mice become paralyzed, the signal creeps up the spinal cord into the brainstem. TSBO PET, on the other hand, we don't really see that kind of clear signal delineation because of the lack of specificity there. We also show in relapsing remitting MS that we can detect disease progression. So as we get remission, that signal goes down. And then as we get a relapse, the signal comes back up again in the spinal cord. And then the last thing I want to show about TREM1 is that we're able to detect a treatment response. So this is very important, using fingolimod, as Dr. Cleland had already mentioned, FDA-approved drug for MS. You see the signal in EAE before the mouse is being treated in the spinal cord, and then when we treat that mouse, the signal goes away in the spinal cord. So very nice as a potential way to monitor therapy. And this is collaboration with the pathology department, Dr. Hannes Vogel. You can see in human MS brain tissue, those brown splotches there are TREM1 positive myeloid cells. And so this is a marker that is relevant not only in mice, of course, but most importantly in humans in the clinical setting in MS. And so we're hoping to translate this to the clinic soon. And then just lastly, as our, just a little bit on B cells, this is we've been working on developing a CD19 targeted PET tracer to really capture a pan B cell marker. Because B cells are very much involved in driving MS. And there's been a lot of successful treatments. Actually, the only treatment approved for primary progressive MS targets B cells. And we want to understand more about which patients would benefit from those treatments and which ones wouldn't, and why maybe it's not working in some people. So here's an example of an image with a CD19 PET tracer that one of my grad students, Samantha Reyes, has been collecting. And you can see signal in the spleen, lymph nodes, very clearly. And it's also in the CNS and the bone marrow. Here, a treated mouse, you get rid of that signal in the spleen and bone marrow, and you think, wow, that's great, it must be cured. Unfortunately, these mice are still all paralyzed. So what's going on? We show that, in fact, there is a decrease in signal in the brain, particularly in the brainstem and cerebellum, but it hasn't been fully decreased. So we dove into that a little bit more, and we see spleen, using flow cytometry, B cells are gone. Great. And actually, what they would do in the clinic is just measure blood and show, okay, we've depleted B cells in blood, it's good. But when we look in the CNS, we see in the brain and in the spinal cord, there's still actually B cells. We haven't got them all. And then we did some staining, and we showed that, in fact, yes, you can see in the meninges, we're pretty good at removing B cells. It's more accessible to the treatment. But in the parenchyma and in the ventricles, there's still B cells there. So we need to get better at that. And I think the exciting thing about this strategy and other strategies like it is it's shedding light on why therapies may not be working, and it will help us to optimize these treatments. So in summary, I showed you a little bit about TSPO PET and how it's been shedding light on what's happening in a disease like ME/CFS with all that muscle signal and bone marrow signal, so it's really opening up a new area. We're developing more specific and selective tracers for looking at microglia and macrophages and their dysfunction in the CNS that we're excited about. We're also looking at adaptive immune cells. And so we'll be continuing this work. And I'm happy to take any questions. I just wanted to acknowledge again Sam and Runa. We miss them. And thank you all for your attention.
How do you separate out the effects of the chronic fatigue on the muscles from the disease itself? Because you're going to see just from less activity changes, perhaps.
Right, so you're thinking activity changes, like how do we sort of limit that before or make sure that people have different...
Looking at the disease process and not
right.
a consequence of the inflammatory...
Right, that's a great question. So there, we're pretty strict about, even with the controls, we say no vigorous exercise for like 2 weeks before, and then we talk to people about limiting their activity. For ME/CFS folk, they already are quite limited. But then the other thing is we have a lot of ways to score their function. It's sort of, there's a metric called time that your feet are on the ground. And so we record all of that and understand how much time people are engaging in any sort of physical activity. So we try to correlate and control for that as well. It's a great question.
Sequential studies early in the disease and later in
Yes.
the disease to try to separate out cause.
Yes, yes, and I'm so happy you raised that. It's something that the community is really interested in right now. There seems to be a separation between the early ME/CFS and the late. So anything 5 years post, there seems to be different types of immune responses. Yeah. Exactly. Yes, no, great points. Thank you. Love to talk more about that later.
Great. Thank you so much, Dr. James. So next up, it's my pleasure to introduce Dr. Anna Wu. She's professor and chair of the Department of Immunology and Theranostics at the Beckman Research Institute at the City of Hope, and also the Fouad Kandil Chair in Diabetes and Metabolism Research. She serves as the co-director for the Center of Theranostic Studies at the City of Hope, and fortunate for us, she's also an external faculty member of the molecular imaging program at Stanford. So she's going to be talking to us about visualizing immune responses using ImmunoPET.
Perfect, thank you. Yes, so I was actually asked to talk about ImmunoPET and clinical translation, and I'm thinking about how to frame the talk. What I really came back with was this concept of push and pull, and actually it relates very much to Sam's philosophy, and you'll see that when I get into that slide. And actually, this is a story, not just of current state of research, but how we got there, and I think throughout you will see Sam's influence. First of all, these are my disclosures. And here's what I mean by push and pull. Investigators in the lab, we have all these cool ideas, and especially around Sam. You know, he said, oh, go for it. Try this crazy cool idea. Cool ideas, cool technologies, cool new models, very exciting, very fun. And we do all this work in the laboratory to develop agents, test them on cells. We bring them into mice. They look really promising. Oh, this is terrific. Let's try this in patients. You know, that's the one side. And I made these slides before all of the comments yesterday, but it just drops right into the influence Sam has had in how I work. But you have to think about the other end, the pull. If we really want to have an impact in the world, we've got to think about what is that unmet clinical need that we're trying to address. And there's multiple stakeholders. And look at the first one on the list, the patients. So throughout, you know, with Sam's influence, I've always also been focused on what can we do for patients. That's what matters. So multiple stakeholders, the patients, physicians, pharmaceutical and other companies, thank you all for coming. CMS, Medicare, insurers, and, you know, in the end, and again, coming back to access, we've got to have the insurers, the payers on board to make this available to patients everywhere. So it really also becomes a question at some point of what can and should we take to the clinic. So that kind of will frame what I'm going to talk about today. First of all, so I started my career at City of Hope under Art Riggs, who was well known for inventing recombinant antibodies. And so I was, actually, he didn't want to work on antibodies anymore. He wanted to work on chromatin structure and methylation epigenetics. So I ended up inheriting his antibody engineering program. But I've always loved antibodies. I'm going to spend a little time on this slide, not go through every word on it. But I love antibodies because of the diversity of the immune system. The mammalian immune system has incredible diversity, specificity, and affinity. You can get antibodies pretty much to anything. You know, there are some exceptions, but to me, it was also a platform. Only the tips of the native antibody are involved in target recognition. The rest is very standard. We have constant regions, we have framework regions, so that part's always the same. And so you can predict the properties of antibodies. You know, and you can mess with them, but you still have something that's going to behave pretty much as you think. You can stick a label on or a dye, and you still have something that has fairly predictable properties. And they're malleable. We're protein engineers, so we can actually tailor these antibodies to suit whatever ultimate purpose we're interested in. Translatable, and we have this tremendous success. You know, I started a long time ago, but now you look at you know, the drug approvals and what pharmaceutical companies are working on and what's working. I mean, it's taken this long to get to that point in terms of our knowledge and capabilities and infrastructure to be able to have this, but it is available. There's infrastructure. You can manufacture antibodies. You can take them into the clinic. It's really straightforward, you just have to have money. Feature or bug, this is Stanford, we have to talk about features and bugs. They have a long circulating half-life, which is great for therapeutic antibodies, but I'll show you not so great for radio-labeled antibodies. And then a bug is that we're limited to cell surfaces, but there's still a lot of really interesting stuff going on at the cell surface. So our question, even at City of Hope long, long ago, kingdom very far away, can we use tumor-specific antibodies for radionuclide delivery in mice and, importantly, and in patients? So that was the question at City of Hope. And so I started working with collaborators on these anti-CEA antibodies. We started with intact mouse antibodies. We actually put a mouse antibody into patients. Then we moved on to a chimeric antibody. But you know, the challenge, so here's the pull-push part. Okay, so the challenge is that mouse antibodies are not ideal to put into a patient. They're foreign proteins, and they have these poor kinetics if you want to do a radio-labeled antibody. So early on, in addition to making chimeric antibodies, we went on to do humanized antibodies and fully human antibodies, so they're much more suitable for use in patients. But also, what our lab focused on was making engineered antibody fragments for that accelerated clearance, which I'll talk to a little more detail in the next slide, but also we can control the organ, or primary organ of clearance, either to kidney or liver, depending on what our final application is in the patient. Do we want to image something in the pelvic region? We want liver clearance. If we want to look at liver mets, we want something that clears renally. We can control all of that through protein engineering. And then I won't talk about this much at all, but we can also do things such as introduce sites for site-specific conjugation and radio labeling. And I'll just say that when it comes to a clinical product, if you've got something more well-defined, it's a really much more straightforward process. So here's a little more detail about how we can use these engineered fragments for imaging and for therapy, what the advantages and disadvantages are. And so you'll see for imaging purposes, we went with these smaller fragments. And as you can see, rapid target uptake in the blue, for these mini-bodies and diabodies. Fast blood clearance, this is the key. Gone in 24 hours for these mini-bodies, gone within 3 or 4 or a few hours for the diabodies. We've actually labeled them with F18 and labeled and imaged in mice. It works in mice. And then that does mean for imaging purposes, we get high contrast at an early time point after injection, rather than having to wait a week or so, which is about how long with a whole intact antibody you typically have to wait in order for all that circulating activity to clear. And this is also important for therapy. I won't talk to you at all today about theranostics, but that is the other major part of our program. Even back in my early days at City of Hope, we were taking these chimeric anti-CEA antibodies, labeling them with indium first for imaging in patients, changing the radioisotope to a hard beta emitter, yttrium-90 for therapy. And it didn't have a buzzword then, but that was theranostics. And so we're finally getting back to that again. So for therapy, we go with the larger fragments, just in brief, and we actually do want liver clearance because we want to spare the bone marrow and the kidney from absorbed dose and radiotoxicity. But that's a story for a different talk. We're going to talk about ImmunoPET. So ImmunoPET came about because of Sam. It started with Mike Phelps, who I was at City of Hope, and he invited me to UCLA to do a sabbatical with this brilliant young assistant professor, Sam Gambier. And so a number of things were converging. One is that, fortunately, back in the old days, there was really only fluorine-18, carbon-11, 2-hour half-life, 20-minute half-life, not compatible with antibodies. But slowly, people were coming out with better accessibility of positron-emitting radionuclides with longer physical half-lives. So copper-64, iodine-124, these are things that we started using on my sabbatical in Sam's lab. So on the left, there's the broader and broader list of radionuclides. This is the laser, there we go, whatever. And on the right was the product of my sabbatical year. We put copper-64 on the anti-CEA minibody, and you can see that we got liver clearance because that's part of the design. It's supposed to clear quickly to the liver, so we get no background elsewhere. And you can see clear signal in the positive tumor, not so much on the left, on the negative tumor, and of course the autoradiography confirmed that. But I also wanted to comment, this slide also hits on another point that was mentioned so many times yesterday and today about Sam's ability to connect people. Because on the author list for this paper are both Mike Phelps and Mike Welch. And those of you who know that they literally got into a fist fight in a bar once, okay? But Sam brought us all together on this paper, which we could not have done without, everybody's involvement. So that was Sam, okay. We went on and made like a whole cottage industry of taking tumor-specific antibodies, reformatting them to diabodies and minibodies, so that we have immunopet agents for imaging, in this case, a lot of human tumors in mice, but looking at cell surface markers in human cancers. Either same day with diabodies, so I think there's a bunch of these are diabodies, or next day imaging in general with the minibodies. And we kind of coined think of this as in vivo immunohistochemistry, because we're identifying these living tissues and living mice based on their cell surface phenotype. Okay, so that's all well and good. But what about immunology? Because that's what we're here to talk about, at least in this session, right? Okay, so here's the poll. In parallel, there's all this really wonderful stuff going on. Just the birth and development of the cancer immunology field. And just in short, up on the left, you can see sort of the paradigm of how we wish things would happen, that tumor cells die, they release tumor-specific antigens, the dendritic cells, the other antigen-presenting cells, take them up, and they traffic to the lymph node. They have this wonderful conversation with naive T cells and B cells and helper cells, and you get cytotoxic T cells that are specific for those tumor cells that get back into the tumor and wipe them out. That's what we wish would happen, and that's what so many immunotherapies are directed towards. The checkpoint inhibitors, bispecifics to recruit those T cells directly, immunocytokines to change the microenvironment, cell-based therapies, CAR Ts, vaccines that theoretically, or in reality, generate a vaccine response in the tumors. So great promise, but there are challenges, diagnostic challenges, and there's tremendous heterogeneity of both tumor biology and immune responses. We all know that. That really is one of the huge challenges of cancer. It's constantly evolving, and different lesions and different cells will evolve in different directions to escape whatever therapy you're trying to apply. True responses versus progression. Dr. Lim spoke this morning about pseudoprogression. Here's an example here of pseudoprogression where the patient's disease looks worse first if you're treating with immunotherapy, and it takes weeks or months before you can determine whether or not there's a true response. And that's because the immune cells are getting in there and making the tumors look larger, and they stay very metabolically active because they do the same metabolic shifts that tumor cells do. Biopsies can provide tremendously detailed and complex information, but there are sampling limitations. I'll talk a little more about that later. And they're invasive, and you can't always do the biopsies you would like to have. And they're usually, when patients have advanced disease, you're not going to biopsy every lesion, even though we know that those lesions can have very different biologies. And blood sampling, again, you can do incredible profiling, but I will point out that only 2% of your T cells are in your blood. The rest are in your secondary lymphoid tissues, or hopefully in your tumors. They're not in the blood, so it doesn't tell you everything that's going on. And then, of course, you have clinical challenges, real but modest response rates. Once you get beyond the core, very responsive, renal cell melanoma, small cell lung cancer, non-small cell. So patient selection can be an issue, especially when we want to get to these other colder tumors. Toxicities, the immune system can overshoot, and there are a lot of toxicities associated with that condition. And then I think a big challenge is rational development combination therapies. We know we need combinations, but there's so many possible combinations, not just within immunotherapy. When you start adding chemotherapy or radiation therapy, it just is an incredibly complex problem, and there aren't enough patients to do the clinical trials that we need. We need better ways to figure out what's working. So that's the pull, the challenges in immunology. And actually, my good friend, Tony Ribas, who's a melanoma immunotherapist at UCLA, came to me one day early on and said, how can we distinguish tumor cells from activated immune cells? Getting back to that issue that conventional imaging isn't a perfect tool. And then he mentioned, of course, as we've also heard earlier today, the CD8 cytotoxic T cell is the actual drug of interest. So when we treat a patient with a checkpoint inhibitor, if you throw that onto tumor cells in a test tube, they'll laugh at you because it's an antibody, it's food. You have to have the cytotoxic T cells on which the checkpoint inhibitor acts, and it's the cytotoxic T cell that actually kills the tumor, right? So we need them. So Tony asked me, how can we distinguish the tumor cells from activated immune cells? I said, well, of course, we'll use antibody imaging, because there's all these wonderful cell surface CD markers for lineage, differentiation, activation state, checkpoints, et cetera. And we just need to take either the anti-mouse or the anti-human antibody, reformat it into a fragment, label it, and we have a PET tracer for these immune cells and also potentially their functional state. So we've done a bunch in the lab, but I'm going to focus the rest of the talk on the CD8 cytotoxic T cell, because those killer T cells are the ones that are essential that we really, really need to have. So this is an early study with Tony Ribas' mice looking at a syngeneic tumor model, CT26 colon cancer in the mouse. The mice were treated, the tumors grow, the mice were treated, this really doesn't work, with anti-4-1BB, CD137 antibody. When you treat these tumors, will disappear, they'll resolve over time. But what you can see is if you grab them here at day 15 and stain them for CD8, you can see a really, really strong CD8 T cell infiltrate in those responding tumors. And we can image it. So on the left is an untreated mouse. First of all, we're using a small fragment of cyst diabi that clears to the kidney, so that's all just clearance to the renal clearance. But you can see a large tumor, a lot of diffuse activity in MIP, and in transactional view, you can see the center of the tumor is cold. There's some activity around the periphery. But in the treated mouse, you can see the tumor is smaller, and in cross-section, you can see it's hot through and through. But you can also see, and this is an agent that acts systemically, the entire immune system of this mouse has lit up. The spleen is huge and bright red. The lymph nodes are much more bright and visible, and especially the ipsilateral draining lymph nodes. And I just want to make this point now before I forget. That's the beauty of doing PET and whole body non-invasive imaging. The immune system is a system. We can look at the whole body, not just, where we can stick our biopsy needle. And I think that's especially important in these applications in immunology. And Michelle gave us a wonderful example of how much we can learn once we can start to look at immune responses in the whole body. Okay, and just a control to show you that the signal is specific. So We've gone on and done a slew of collaborations using the mouse-specific agents, including, I saw her earlier with Kathy Ferrara and her group, and also Hangjin Dai and his group. So we've had at Stanford, so we have some wonderful collaborations with you all. If you're interested, let me know. We can send you the agents for your mouse work. But look, in oncoimmunology, we've done collaborations with colleagues doing a variety of different therapies, checkpoints, adoptive cell transfer, combination oncolytic viral therapy, bispecific antibodies, external beam radiation therapy, looked at CD8 T responses in those mice, but also their non-oncologic applications, because this is the immune system, stem cell transplantation, graft versus host disease, infectious disease. So it's really much broader than just oncology or neuroinflammation, although that's what we're focused on. Okay, translation and commercialization. So is Chris here? He's got to be here someplace. So if you were here yesterday, you heard this story. But anyway, to be a little more specific, if you're in an academic setting, it's a challenge to really bring something into the clinic. You need a multidisciplinary team with so many different expertise, including highly engaged clinicians, because they're the ones that have to recruit your patients and inspire them to join an imaging study, which has no therapeutic benefit to them at all. It's a real challenge, and you've got to have the clinical team that can execute those clinical trials. You need to be able to produce the agent, you need to be able to scale up, you need radio label at clinical grade, you need the regulatory experience to help you file that IND. Thank goodness they're all electronic now, because the last one we did a paper IND, it was 2,000 pages. And grant funding can only, this is one thing I learned at City of Hope, grant funding can only support you through maybe a phase 1 trial or maybe a very early phase 2 with maybe 20 patients at most. If you want to get beyond that, you have to have involvement with industry. You need those dollars. So thank you all of you industry folks who are here supporting the field and really moving it forward. And I also learned, again, I want to echo, you need to address an unmet need, to inspire people to invest in your idea. You need commercial support if your ultimate goal is to have broad availability. You need the intellectual property. So please, students and postdocs, make sure you file those invention reports. But I know that Stanford does a good job making sure you do that. And as I mentioned earlier, and others have said, imaging is a tough sell in clinical trials, so you got to have the right people. But anyway, these are some pictures from the early days of ImagineAb, and here's our fearless co-founder, Chris, helping set up the labs and our first 2 employees with our makeshift bench. And this is myself, and so that's Chris and Rob Ryder and I were the founders. This is Rob and I in a snowstorm in Boston trying to meet with venture capitalists. But anyway, here's the key picture. An early supporter of this entire process was Sam. He was on our scientific advisory board for many years, and even though he wasn't directly involved, he was a tremendous supporter, and as Chris mentioned earlier, a critical supporter in 2 ways. One, we needed his support, and 2, we needed his criticism to keep us in line and keep us moving in the right direction. So again, Sam's fingerprints are all over this. So quickly to the, only 1 minute left, 1.5. Okay, I'll blast through this. We made a clinical agent called crefmurlimab burdoxam. It works in patients, so here is a melanoma patient showing biopsy confirmation of the CD8 imaging in patients. This is the phase 1 trial. It was published, 15 patients. It allowed us to establish basically our RP2D. Now we're using 1.5 milligrams of protein labeled with 1 millicurie of zirconium-89, and we're imaging 24 hours later, and we can absolutely see CD8 T cells in the patients. And we confirmed this through some of our phase 2 studies. Our phase 2 studies look at patients who are undergoing standard of care immunotherapy. They have a baseline scan pre-treatment, and then 4 or 5 weeks after they start checkpoint inhibitor therapy, we scan them again to see if there's a change in their CD8 T cells. I'll point out one of the challenges, which I didn't realize until you start working with clinicians. When you do the biopsy, the patient is prone, but when you do the scan, the patient is supine. And then you stick the skinny little needle in. Sometimes the uptake is heterogeneous, and first of all, how do you find the right tumor, the right spot, and then correlate that with your image? So a lot of challenges along the way. But here are examples in renal cell and non-small cell lung cancer, both pre-treatment and on treatment, where you see an increase in the CD8 PET signal and in the corresponding biopsies an increase in the CD8 immunopet. So there is a good correlation. It's not perfect, but we wouldn't expect a perfect correlation. because of sampling limitations, because of heterogeneity. And then there's this broader question, is CD8 immunohistochemistry really the gold standard? So I'm going to wrap up here. I did want to show you that this is relevant across indications. So here's immuno-oncology on the left. In the middle, we were very fortunate to work with Simon Cherry and his group that have the whole body scanner up at UC Davis. These are patients who've recovered from COVID infection 4 months later. But look, their bone marrow is still highly active and full of T cells, CD8 T cells. These hopefully are your memory T cells that are helping you to have sustained immune response and protection against COVID. But we never have been able to see this kind of response, or the state of the immune system before. And then on the right is an autoimmune disease. And actually, this kind of reminds me of Michelle's slide, because this is inclusion body myositis, where CD8 T cells infiltrate muscle. And you can see very clearly the CD8 PET signal in the patient's muscles. So we're able to visualize all of that non-invasively. So summary in future, I'm not going to spend any time on this. But just that we do believe we are imaging CD8 T cells in patients. I'll just point out this one investigator-initiated study that I'm involved with at City of Hope that I'm very excited about, which is looking at CD8 T cell responses in response to radiation therapy. So these are patients undergoing standard of care radiation therapy. We image them before and right after they finish their 5 cycles of SBRT, and are looking at changes in the CD8 T cells, not just where we irradiate, but also in other lesions in the body. So we're starting to explore that question. What is the immune response to radiation therapy? So I'll stop there, but I think I pointed out the push that's important, but also the pull that's needed to really bring things through into the clinic. And if we put it together right, we hope to have an impact on patients' lives. So I'd like to thank my labs, our industry partners, all of the people who participated in the clinical trials, and for Sam, who inspired and I feel has been watching over this entire process. Thank you.
You mentioned collaborations. So how do you go about ensuring that academic and/or clinical collaborations are effective? What do you do to nourish them, and what percentage of them end up being long-lasting from your perspective?
Yeah, I think the way you nourish the academic clinical collaborations is you have to start talking to the clinicians right from the start and really understand what the challenges are and what the problems are. And that's one of the reasons I was so happy to go back to City of Hope, was to get more directly involved in clinical trials and see what the hurdles are in terms of recruitment and scheduling. And so a lot of it is just listening to them, but it's really driven by what are the questions they need to answer, but then learning from them as well the hurdles of getting it all done, and finding collaborators who really want to help bring new processes forward. So, it's just a multifaceted challenge, but you find the right people to work with. That's the key, and then you can make it happen. Does that answer your question?
Yes, that's great. I think we learned yesterday how Sam did it, and that was constantly giving.
Other questions, or
Any other
are we ready for
questions?
lunch?
Oh, okay, yes. We're going to break for lunch, but not before. I just want to thank our 2 amazing speakers for the phenomenal translation efforts. Thank you for sharing your work, and yeah, please enjoy lunch. We'll break for an hour.